Hi everyone. Just a second, let me just check this. Yeah.
For the nice fit.
Okay, yeah, fine. So there was some problem. Now it is solved. Okay. So let's see this. So we'll discuss about the different, the plant-water relationship, that is, relation basically. So that is with respect to, imbibition. Then there is osmosis, the diffusion, active transport, passive transport. So different terms along with, conditions like turgidity, flaccidity. So we will see all that in today's session. So this is the syllabus and the scope of syllabus that you need to follow. Before you start the chapter, always make sure that you are referring these. Okay, so now make sure that you cover each and every topic which is mentioned in here. So this is there is nothing that is gonna come out of this syllabus or a scope of syllabus basically. Okay, so all you need to be is thorough with these concepts which are mentioned in here. All right. So before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. All right. So join with us in Telegram. So that is for homework and class notes. You can join the channel Vani Kari ICSE Class 9 and 10 Biology. And this is the Telegram channel, that is Unacademy ICSE and ISC both. So this link you can find in the description box. Alright, so go to the description box, click on this link and join the channel. Also, we conduct special classes on Unacademy platform, which are absolutely free classes. Okay, but here the certain percentage of topics will be covered, but you have access to the classes of all the educators once you download the Unacademy Learners app. So learn from your favorite educator for free, engage with educators via poll. So here we'll be conducting a quiz, so you will have access to the quiz. So that is once you download this one, so that is Unacademy Learners app, and start attending the free classes that are there. So a lot of them will be conducting a quiz as well, so you will have access to the quizzes, because you need to use the code RNYT if it is asking you a code to unlock. So you can use this code to unlock many of the free products that are there on Unacademy platform. All right. So clear your doubt in the live special class. So there is a question tab where you can type in your question, attach an image and send it to the educator during a live session, so they'll be clearing your doubt. So for the top special classes, you can visit the link in the description. All right, so you can visit the link in the description. So there you will find the a special class link. Okay, so now these are the classes that I was talking about. So these are plus classes. So learn live from the comfort of your home. Unlimited access to all the courses will be there. Then top educators of India are going to take the classes in one platform, that is Unacademy platform. There is a regular doubt clearing session that is done. So that is after about every 3 sessions. The fourth session will be your doubt clearing session. Okay, so whatever doubts you have related to the topics that are previously covered, so you can clear your doubt in this doubt clearing session. Then complete syllabus will be covered, mentorship and guidance is given, study material is given, the practice tests are also given, and with that you have a live test series and the batch courses. So batch courses here, basically we have a batch in English, we have a batch in English. So once you take the subscription, so you will have access to all the batches. You can take the subscription with these, and depending on your comfort you can go for any of these batches. Just a second. And then there is daily practice section is there, okay, where you can practice on a daily basis. There is also a weekly mock test series where the analysis is also done. Live quizzes are also conducted by in many special classes, like a lot of educators keep comment, conducting quizzes, so you can go for these. And other than that, there is daily MCQ and subjective test series will also be there. Okay, so now this is with respect to Unacademy plus subscription pricing. So since this is the very beginning of your class 10 and also very beginning of your academic year, so you can go ahead with 12 months of subscription. So if you are going for 12 months of subscription, so basically you are getting class 11 and 12 subscription also absolutely free along with this. Okay, so if you are planning for to take a subscription with respect to class 10, so maybe you can go with 12 months because this is the beginning, or if you just want to try, so maybe you can try with 6 months, 9 months, or 3 months depending on your comfort. So those who are planning for your higher classes as well, so maybe you have an option to go with the higher months of subscription as well. Okay, so you can plan accordingly, and don't forget to mention the code Vani YT, so that you need to refer here in the gift box, so that is Vani YT, where you will be getting 10% discount on the subscription. All right, so 10% discount you will get only with the referral code, so there is a reduction in cost that can be seen. Okay, so from this chapter, that is absorption by roots, we'll be discussing about the processes that are involved. So those processes are mainly the imbibition process. Okay, then basically the water plant water relation, what exactly is the a mechanism that is occurring in order for the water absorption. So here this is with respect to the plant water relation. So I'll just go with this. Okay, so absorption and conduction of water and minerals. So how exactly this occurs? So this is the absorption by the root. So what exactly is getting absorbed? It is the water and some of the mineral nutrients that are being absorbed here. Okay, so there is water absorption, there is a mineral absorption. So let me just check. Yeah. So here in this case, when the absorption of water occurs by the root, so it is not just the water that is moving in. So along with the water, so there are mineral nutrients are also that are taken inside. Okay, so for that process we have a lot more processes that are occurring. So let's see what exactly happens here. So the entire mechanism of water absorption and minerals from the soil by the roots. So this is basically through the thickness of the root and subsequently the upward conduction. First the water has to be taken in depending on the thickness of the root, and then whatever water has been taken in, so this has to be moved up. So because in taller plant the water is required in the upper plants or upper parts of the plant body as well. All right, so when we say that, basically you can see the xylems are the ones which are transporting the water. All right, so with respect to that, there is a process called as imbibition, diffusion, osmosis, active transport, turgidity, and flaccidity. So all these different processes are actually responsible, so with respect to the movement of water that occurs. Okay, so now when we mention that, so upward conduction is gonna occur, so that is against the gravity the water is moving. Okay, so how exactly water is moving? So water movement you can see against gravity. So for that there is a pressure required. Okay, so what is that pressure required? So just the xylem is not sufficient, so that pressure is basically a suction force or a suction pull. Okay, so there is a suction pull that is created, and that suction pull is the one which is making the water to get absorbed. Okay, and there mainly the transpiration plays a role, which you will study in detail. So there the transpiration plays a major role. All right, so this can be with respect to the suction pull. All right, so now here the absorption and conduction of water and minerals. So you can see these are the processes, and let's have a look at each one of them one by one. So imbibition is a phenomenon. So what is that phenomenon? It is not just to a living cell. It is, it is respected to for both living as well as dead plant cells. So that is, the living cell is also absorbing the water by surface attraction. The dead cell is also absorbing the water by a surface attraction. So now let's say, now this is the surface of a cell, and here you can see basically, the water is present. So because of the surface attraction, so there is a movement that can be seen. Okay, so water can move in. So this can happen in dead cell, so this can happen in living cell. So this definition will be asked many a times, like, what exactly is imbibition. So just make sure that the surface attraction is the key term. Then, living or a dead plant cell is again a key term. Okay, so this is a phenomenon by which the living or a dead plant cells absorb water, so that is by surface attraction. So now here, there is a cellulose which is hydrophilic, then they imbibe water, so that is they are taking in the water or the moisture, and they are going to swell up. So examples we can say dry seeds are there, wooden doors might be there. Okay, so they, like, usually in rainy season if you observe. All right, so one second. So this one, in rainy season, what do you find? So basically you cannot close your door. Okay, so doors are not closed easily. So reason is what? Okay, so that is, you can see a clear swelling that has seen in the doors. Okay, so doors are not closed easily. So what is the reason? Because the doors, when we mention, so you can mainly find the dead cells, and since it is a rainy condition, so the doors are coming in contact with the water, and because of that there is a imbibition process that will be happening. Okay, so that is a surface attraction you can see between the dead cell and then the water. So due to the surface attraction, the dead cells are gonna swell up. Okay, so these will swell up, and that's when you feel a difficulty in opening or closing a door, especially. So that is because the water has been imbibed into these dead cells, and that will lead to the swelling up of the cell. Okay, so this can be the example, and other than that, so swell up on contact with the water. The dry seeds also can be the example, so that is soaking the seeds in a water. Then, and on exposure to the moisture, so this is also done. Then due to the imbibitional pressure, the seed coat is gonna rupture. All right, so that is in case of a germinating seed. So this is also an important force in the ascent of sap. So how exactly ascent of sap occurs? So as there is a seed coat rupture, so germination that occurs, so you can see the movement of the cell sap. So that is water and minerals which is present in the vacuole. So there you can see imbibitional pressure is acting. So due to imbibitional pressure, the seed coat is rupturing. Okay, And in case of, that is mainly in case of germination, so germinating seeds, and ascent of sap. So that is what exactly is ascent of sap. So I'll just explain you here. Okay, so let's say this is one cell. Okay, so here is another cell. So now cell to cell communication has to occur. So cell to cell transportation of water, mineral has to occur. So in that case, so this is a hypo condition. So this one is a hyper condition. Okay, hypertonic solution I'm talking about. So this is also a hypertonic. This is also a hypertonic. Okay. Now what will happen? The water will move from the hypotonic solution to a hypertonic solution. Okay. Because this one is a dilute solution, whereas this one is a concentrated solution. So now what happened? As the water moved here, that itself we call as ascent of sap. Okay. So now this became hypo. All right. So now surrounding again still it is a hyper itself. So now water will move from here to here. So this became what? This became hypo again. And now this side, as the water quantity is reduced, so there you can see the water movement that is occurring. So this was earlier a hyper itself, which later on has become hypo due to the water entry. So same way, now this was a hyper earlier, and same way now hypo solution, that is water will enter. So this is gonna turn hypo. Okay. So in that case there is a alternate turgidity and a flaccidity you can see. Okay. So with respect to alternate turgidity and a flaccidity. So this is with respect to turgidity and flaccidity. Yeah. So alternate turgidity and flaccidity are the ones which are making this root pressure. Okay. So that is called as a root pressure. So which is conducting a water in a small plant. Okay. So that is one of the things. So now moving on to this diffusion. So what exactly is diffusion? So you all know the definition. It is applicable to solute. It is applicable to solvent. It is applicable to gas or a liquid. So basically everything it is applicable where you can see the free movement of molecules. So it can be gas, it can be water molecules. So free movement of molecules you can see. So that is from the region of their higher concentration to the region of their lower concentration when the two are in direct contact. So in that case, let's say here they have mentioned a activity. So a sugar cube is kept. So let's say we are taking a beaker, and here we are keeping a sugar cube. So if you have visited any places, so basically you can find bigger sugar cubes. Okay. So you can just try keeping it in one corner, and or you can also try it with the potassium permanganate as well, but this is the easier one that you can try even at home. Okay. So now the sugar, the dye, or the potassium permanganate, so they will slowly dissolve. So they'll start to dissolve and spread in the liquid. Okay. So they'll start to spread. So now that is how you can see once they start to spread, so eventually the molecules of this substance, so now this is a sugar. Okay. So all the sugar substances that are there, so they will start to get move from their region of higher concentration to their region of a lower concentration. So with that, so as you can see, slowly dissolves and spreads in the liquid. Eventually the molecules of this substance are diffused or distributed uniformly throughout the water. So now you can see that whole water has been spread with this sugar. So now if you try to drink this water, so this will be sweeter because the sugar has been dissolved there. Okay. Any doubt till here? Alright, so with respect to that, we have the next process here, so that is osmosis and osmotic pressure. So now what exactly is osmosis or osmotic pressure is? So we'll discuss about the osmosis now. Diffusion is without any membrane, okay? So there is no involvement of any membrane. Same as that of a diffusion, osmosis is also same, but there is a barrier, so that is, a semi-permeable membrane is there where it allows only the water to enter freely, that is solvent to enter freely, but the solutes are not allowed inside. Okay, so osmosis is basically movement of water. Okay, so that is water molecules from their region of higher concentration, that is dilute solution, to their region of lower concentration, so that is a concentrated solution. Okay, so that is across a semi-permeable membrane. So what is most important to note here is the water molecules move from their region of a higher concentration to their region of a lower concentration. So that is a dilute solution to a concentrated solution. So what is mentioned here? Inward and the outward osmosis is mentioned. So what is inward and outward osmosis? So now here is the cell. Okay, so in this case the water is moving inside, so this we call as end osmosis. Okay, so let's say here is the cell and the water starts to move outside, so that we call as a exosmosis. So what is the condition why there is a water moving in or why there is a water moving out? So why this is happening? So inward diffusion of the water through a semi permeable membrane when the surrounding solution is less concentrated. So this one here is a dilute solution where you can find the more of water concentration. Here inside the water concentration is less, and hence you can see the water will start to move from outside to the inside. So here what is happening in case of okay, so here let me just say, so once the water moves it, so this is gonna swell up, okay, and that condition is basically called as a turgidity. So turgidity of a cell, when we mention basically the Solon condition of a cell. Okay, so same way now here concentrated solution is placed, that is surrounding is concentrated, and water here is actually more and water here is actually less. So what will happen? The water will move from their region of higher concentration to their region of a lower concentration, and with respect to that, so this is the outward diffusion of water through a semi-permeable membrane, which is basically a cell membrane itself, and the surrounding solution is a concentrated one. So this will result in the shrinkage of the cell. So after this process, basically you can see the cell has shrunken. Okay, so shrinkage in the cell you can see, so that is because of the exosmosis that happens. Now osmotic pressure, when we mention, so this is the minimum pressure that must be exerted. So this is to prevent the passage of the pure solvent. So in that case, now a pressure is required to stop or to nullify a osmosis, we can say. So osmotic pressure is the minimum pressure that must be exerted to prevent the passage of the pure solvent into the solution when the two are separated by a semi-permeable membrane. Okay, so when two of them are separated by a semi-permeable membrane, so there is a complete, you can see water keeps on moving inside. So in order to prevent that water movement into the solution, there is a minimum pressure that is required to prevent the passage of the pure solvent, and that is that is occurring across the semi permeable membrane, so that we call as a osmotic pressure. All right, so now osmotic pressure of a solution is a measure of its tendency to take in that is water by the osmosis process. Just a second. Where did this go? Yeah, here it is. Alright. So in this case, osmotic pressure of a solution is basically measure of its tendency to take in water. That is by osmosis itself. Okay, so now we know this is by osmosis itself. Now you can see the water that keeps moving inside. Okay, so this is with respect to the changes that are gonna occur. So some of the examples are given. So this is for an animal cell which is placed in a isotonic solution. So there is no net movement, or whatever amounts of water are entering, whatever molecules of water are entering, so same amount are leaving here. So water entering the cell equals the water leaving it. Okay, so the same thing with respect to plant cell. Okay, so water, no loss or gain by the gain of water by the cell, because it is kept in a isotonic solution. That is, iso is the same. Yeah, so I was just discussing about this. So what exactly happens is, when it is kept in isotonic solution, so there is no loss or a gain. So because it is the outside solution is also same. I mean the same concentrated. The inside solution is also same concentrated. So when a same concentration is there, so there is no net movement, we can say. Okay. But now here with respect to placement of the cell in a hypertonic solution. So you can see what has happened here. One second, I'll just change the cursor color. Yeah. So now you know hypertonic solution. So that means it is a concentrated solution. So when it is kept in a concentrated solution, so basically you can see that the water entering the cell is less than the water leaving it. Okay, so in that case the water will start to move out and very less movement of water is occurring. So because of that we can say that the exosmosis is occurring, and you know the cell is gonna shrink. So this is seen in case of a hypertonic. So same with respect to hypertonic in case of plant also. So you can see that the cell membrane is getting separated from the cell wall because of the continuous water movement that occurs out, since it is a concentrated solution in the surrounding. So what will happen, the water will start to move out. So this is the water will start to move out, and because of that you know the cell membrane is getting plasmolized. So that is the cell wall and the cell membrane will get separated. Okay, so they'll get separated, and you know this condition we can say the process is basically a plasmolysis process has occurred, and condition of the cell is basically a flaccid condition or a flaccidity. Okay, so now when it is placed in a hypotonic solution, you can see there is a water that is entering more. So water leaving is very less. So water entering the cell exceeds the water leaving it. So now here in this case what can be seen? Hypotonic solution. So what is hypotonic solution? So now when we mention hypotonic solution, so you know it is a dilute solution itself. So here also it was a dilute solution itself. Okay, so now you know as a dilute solution is there, so basically the water will start to move inside. So that is from their region of higher concentration to their region of lower concentration. So with respect to that you can see that the Turgor pressure is maintained again, and the wall is exhibiting its pressure. So that is a wall pressure. Okay, so now what exactly is active transport? So this is a passage of a substance, so that is salt or ion, from its lower to the higher concentration, so opposite to what happens in diffusion. So this is using the energy from the cell, so that is through a living cell membrane. So this we call as active transport. So what is the meaning of this? Opposite to what happens in diffusion. So why? Because it is always high to low will be there. Here what is happening? There is a movement that is occurring from low to high. Okay. So it's lower to a higher concentration. So to bring something from low to high concentration, always ATP is required. So there is a energy requirement that is always done. So with the by spending energy, like by spending ATP, so what will happen? So basically the things like ions, the salts are taken from low concentrated area to a high concentrated area. So certain nutrients such as ions of nitrates, sulfates, potassium, zinc, manganese, so these and all, they cannot pass through the cell membrane, so that is of the root cells, easily. Okay, because it is semi permeable, so solutes are not allowed. So this is because their concentration is higher inside the root cells. Okay, cannot pass through the cell membrane of the root cells easily. So this is because their concentration is higher inside the root cell, and it is so maintained in order to develop the osmotic pressure for absorbing the water. Now in this way, the concentration gradient of these ions is opposite to that of the diffusion. In other words, to obtain them, so these ions will have to be forcibly carried inward. So these have to be taken inside forcibly. So that is from the region of their lower concentration to their region of a higher concentration. So that is inside, and this requires an energy. So energy has to be spent, then only the ions can be taken. So that is from low to high. So why it is maintained high? So that the water can be absorbed, and that's the reason high concentration of salts is maintained always. Okay, so now every living plant cell, directly or indirectly, when we mention, so they are in contact with the fluid. Okay, so there is one such is the root hair, which is in particular surrounded by the soil water. Now when it is subjected to osmosis, the water will continue to enter as long as the cell sap is more concentrated than the surrounding fluid. Did you guys get this point? So cells are subjected to osmosis, and the water continues to enter as long as the cell sap is more concentrated, okay, than the surrounding fluid. So when a cell reaches a stage where it cannot accommodate any water, so that is it is fully distended, so that we call as a turgid, and the condition is basically a turgidity. Okay. Okay. So here with respect to that, so you can see this is a Turgor pressure. So as the water moves inside, so cellular contents are putting up a pressure on the wall. So that is called as the Turgor pressure. So in order to prevent the bursting of this, so wall is exerting a pressure on the cellular content, and that is called as a wall pressure. So water moves into the cell due to the osmotic concentration of cell content. So in that case, there is a pressure that is exerted in order to prevent the passage of the water. So that is the osmotic pressure. Okay, so this all a plant cell showing osmotic pressure, Turgor pressure, and a wall pressure is shown in this picture. So now you can see this with respect to that. Now whenever the cellular contents are getting separated, so let's say this is a plant cell. Okay, so there is a cell wall, then there is a cell membrane here, and as the water enters, that is endosmosis occurs. So now this cell membrane is gonna enlarge, so this will put up a pressure. So as it enlarges, so cell membrane is gonna put up a pressure on the wall. Okay, so as the water keeps on moving, so that pressure itself we call as a Turgor pressure. And in order to prevent, so since cell wall is actually rigid structure, So in order to prevent the bursting of this cell wall, so basically you can see that the wall also puts up a pressure on the cell wall content, I mean cellular content, the cell membrane content, and that is called as a wall pressure. So there is a wall pressure and then there is a Turgor pressure. And osmotic pressure is basically, a pressure exerted, maybe we can say a wall itself, where it will try to stop the osmosis, because we do not want any further entry of water inside the cell, because the cell is already distended. Okay, so if there is a further distension of the cell, what will happen? The cell is gonna burst open and the cell is basically dying. Okay, so in order to prevent that too much water entering inside, so osmotic pressure will try to nullify the osmosis, or it will try to prevent the entry of water inside this membrane, so that is by exerting a pressure. So same experiment is shown in your book, so that is by introducing a piston, so where the piston is applying a pressure so that the end osmosis process does not occur. So there is an experiment with respect to pressure, piston, where it will apply a pressure on the content so that the end osmosis does not occur. So the cell will remain intact. So turgidity is this one. So what is flaccidity now? So flaccidity, when we mention, so when all the cellular content, so let's say all this part is coming at one corner from here, okay, so that we call as a flaccid condition. So this is with respect to flaccid condition, where you can see a cell membrane is is separated from the cell wall completely. Okay, so this is separated from a cell wall. So this can be seen in a flaccid condition there. Okay, so we saw all the terms. Any of you have any doubts here? So Turgor pressure and the osmotic pressure. So there are different things. So any of you, any doubt with respect to Turgor or osmotic, or no? The rest of the thing, imbibition is clear, diffusion, then there is turgidity, flaccidity. So this is turgidity and flaccidity are conditions of the cell. Plasmolysis is a process where the flaccidity is achieved. Endosmosis or exosmosis also. So endosmosis is where the turgidity can be achieved. Exosmosis is where the flaccidity can be achieved. So that can be with respect to the cells there. Okay, so endosmosis and exosmosis, depending on the water, where hypotonic or hypertonic, always you can see the water movement from a hypotonic solution to a hyper. In case of active, so always a water constant, I mean the solute concentration in the vacuole is maintained high, so that the water absorption can occur easily. And in order to maintain the same concentration, what will be done? So more and more ions are taken, so that is from low concentrated area to a high concentrated area. All right, so if you guys do not have any doubts, I'll just end this session. So before that, let me just say about the batches. So there is a gladiators batch, which is a all subject batch, and the mode of delivery is English here. And you can see PCMB is covered in these subjects. So if you are subscribing for class 10, so you will be getting class 11 to 12 subscription absolutely free. And note down the timings of these sessions. Okay, so introducing community, so never miss on any update from your favorite educator. Then get session notes and practice material. Share your feedback directly with the educator via poll. So link to the educator community you can find in the description, and links for the bugs bounty, that is opportunity to for all the learners to report any inappropriate content. So be the first one to report a particular issue to claim your prize. report any inappropriate content using the form in the description. So Bugs Bounty link also you can find in there itself, so that is in the description. So thank you all. So see you all in the next session. So subscribe to our channel, that is Unacademy ICSE and IC boards. Click on bell icon to get notified about the session. Like and share the video and download the Unacademy Learner app. So this PDF, if you need, I can send it on my community or a telegram. So this is basically what I have taken is from a Selena. Okay, so the PPT which I have made, so this is made from a Selena textbook. So if you have Selena textbook, so this is that is more than sufficient. Okay, and next class will discuss, I think I have assigned a session maybe on genetics, or I'll just let you know. So or you can just subscribe to this channel, so you will understand it, or you will get notified whenever the classes are starting. Okay, all right then. So see you all in the next session. Bye bye everyone. Thank you so much. Take care.